Good afternoon, guys. Let us see about the prions. Prions, they are the infectious agent composed of protein in misfolded form, initially thought to be virus, which replicated slowly within their host, but no nucleic acid found associated with the infectious material. They are shown to be aberrant form of normal cellular protein, which can induce changes in the shape of the normal homologous with catastrophic changes for the host. The normal form of protein called PRPC, while the infectious form is PSP-SC. Stanley Prusiner discovered prion in 1982, won Nobel Prize in 1997 for the physiology of medicine. Prions don't have the DNA or RNA. They are extremely resistant to heat and chemicals. They are very difficult to decompose biologically. They survive in the soil for many years. They affect both humans and animals. They does not produce any inflammatory or the immune reactions in the host. They spread to human by infected meat product, resistant to the ultraviolet standard disinfections and the protease K. The common form of the prion that affect the human is Creutzfeldt-Jakob disease. Nature of the prion challenges for the early diagnosis known as transmissible spongy encephalopathy. Here we can look for the history of prions. In 1730, the scapi in the English sheep. In 1950, Kuru appeared 4 people of the New Guinea. In 1960, demonstrating the transmissible nature of a Kuru and CID. In 1982, Dr. Stan K. Prisoner coined the term prion. In 1986 to 2000, 1,80,000 cattle became infected by BSE. In 1997, scientists identified the PRP gene. Here we can look for the characteristics of the prions. Characterized by the spongiform change caused by intracellular vacuoles in the neurons and glia, containing about 250 amino acid abnormal variants of the normal proteins. They convert normal to abnormal form. No antibiotics can cure the disease caused by prions. Not typical of the prokaryotic or the eukaryotic organism. All that present in this pathogen in the protein is the protein PRPSC, the mutation of the PRPC. The PRPSC is resistant to any form of digestion. It is non-immunogen and do not induce an immune response. Prions, they are not easy to decompose biologically. They are resistant to the high temperature and the disinfectants. Normal protein, which comes under the alpha helix, they have the secondary structure dominated by the alpha helices. They are easily soluble, easily digested by proteases. They are encoded by the PRNP gene in the humans. They are located on the human chromosome 20. They are monomeric in nature. Abnormal protein beta helix, the PRPLC for scapi, some amino acid sequence and the primary structures as a normal protein. The secondary structure dominated by the beta conformation. When PRPSC contacts the PRPC, they convert it to the abnormal form. Insoluble in all but strongest solvent, highly resistant to the digestion by proteases, services in the tissue postmortem, extremely resistant to heat, normal sterilization process and sunlight. There will be no detectable immune response. They are multimeric in nature. Prion biology. For a prion to infect a host, the host must have a recognizable cellular form of that of a prion. The closer the phylogenetic relationship between the host and the recipient, greater the chance for the infection and more rapidly the symptoms occur. The level of accumulation of a prion does not necessarily correspond to the level of disease. Characteristics of infection: loss of the motor control, dementia, paralysis, encephalitis, widespread neuronal loss. TSC disease, the prion disease, human and animal, Creutzfeldt-Jakob disease, Gressman-Stossler, fatal familial insomnia, and Kuru. While in animal they have scrapie, which affects the sheep and goat, transmissible mink encephalopathy, bovine spongiform encephalopathy, and chronic wasting disease, which is occurred in mule, deer, and elk. Prion multiplication, heterodimer model. When a prion enters a healthy organism, it induces the existing properly folded normal protein to convert into the disease associated prion form. The normal form of the protein is called PRPC, while the infectious form is PRPSC. The newly formed prions can then go to convert more proteins themselves. This triggers a chain reaction. The large amount of the prion form, which is amyloid fiber. The prions cause other similar proteins to also misfold, lose their function, and cause the disease. Scrappy, this is a non-conventional disease agent. In 1732, they are reported in UK affecting the wool industry. Geographical distribution is worldwide, except the Australia and the New Zealand, and in developing countries they are also known as radar. The name scrappy is derived from one of the clinical signs of the conditions, itching the sensation in the animals. The clinical signs are the excessive lip snacking, altered gaits, convulsive collapse. It has a long incubation period which is 100% fatal. They cause no immune response. They demonstrated to be transmissible in the 1930 despite lack of inflammation, whether it is camelcated virus. In 1930 to 1950, numerous experiments demonstrated the scrappy agent in the extraordinary resilient it survives. They are 30 minutes of boiling will be taken and the 60 days of freezing should be taken here. They will be comes under the strong formaldehyde. The desiccation is nearly 2 years. They are intense UV exposure should be done. The size of the scrappy agent they passes through the fine filter stays in the solution despite the ultra-centrifugation. In 1966, the electron bombardment experiment demonstrated that if the scrappy agent contains the DNA genome, it would be 1,000 times smaller than the smallest known as the virus at the time. Composition of a scrappy agent. UV damages the DNA or RNA severely, but not protein. Scrappy is resistant to the UV. Date 1960. Scrappy agent is treated with the nucleases, DNA or RNA, digesting the enzymes and the protease. The protein digesting enzyme. Scrappy agent is only damaged by the proteases. Symptoms of scrappy. Symptoms may take nearly 2 to 5 years to appear. The head and neck tremors. Skin itching. As the disease progresses, animals have the high sleeping or the trembling convulsions. The scraping body with the wall. Drink small quantities of the water, often blindness. And they have the inability to control the legs. The good appetite accompanied by the weight loss, excessive lip smacking, and the altered traits. Transmission and the pathogenesis. When understood in the sheep, can transmit a goat if we keep them in the herd of sheep. They can transmit to already present sheep if we keep foreign infected breeds in our flock. They can be transmitted through the race during the pregnancy or at the time of birth from dam through the placenta or the reproductive tract. Diagnosis. We can do the laboratory test by detecting the P or PSC prions in the CNS. We can also do the ELISA Western blotting immunohistochemical method. Control measures like public health control measures such as surveillance, culling sick animals, and banning the specified risk materials. Guru, this is a rare and the fatal nervous system disease, a disease of which affects a man. Trembling associated with the fear of the cold, transmitted by cannibalism in the 4 people in the New Guinea. They have 3 phases. Initial phase abundant with the minimal tranquil ataxia, dysarthria, and the tremor. They have the symptoms like poor condition, difficulty in the swallowing, slurred speech, moodiness, and the behavioral changes, dementia, muscle twitching, and tremors. They belong to the transmissible spongiform encephalopathy. Primarily affects the cerebellum, part of your brain responsible for the coordination and the balance. The diagnosis is nearly neurological examination, brain biopsy, histology, CSF 14-33 protein and the tau protein. Sensitivity 94% and the specificity is 93%. Treatment is there is no successful treatment for the Kuru other than discouraging the practice of cannibalism. Currently, there are no cures for the treatment of any other TSE disease. Brain contaminated with the prions remains infectious even when preserved in the formaldehyde for years. Thank you.